Hi, I'm Nicola. I work for the National Heart and Lung Institute, and today I'm going to teach you about fluorescence and explain to you how we use it in the lab.
What's fluorescence?
If you hold on a second, we'll get to that bit in a minute. So, fluorescence is the property of some substances whereby if you shine a light on them of a certain colour, they will give off light of a different colour. So I'm going to try and explain to you why this is and then give you some demonstrations as to some of the applications that there are in everyday life as well. So first I need a volunteer. Is anyone volunteering?
Yeah. What do you want me to do?
So if you just sit down.
Yeah.
And as I'm speaking, I want you to throw the ball up and down and catch it. So there are these very small things called electrons which have a negative charge. No. And that's going to be symbolised by this ball here, which this gentleman is so kindly throwing up and down for me. So in fluorescence, when the light is shone on these electrons, they get excited and they shift up to an increased energy level, which is simulated by this ball going up into the air. And then they can't stay up there forever, so eventually they will drop back down to their original level, and it's during this process that the light is given off. So we shine a light of one colour, they're excited, they go up, they drop down, and as they drop down, they give off light of a different colour. So that's what's going on when we're shining our lights on things, which I'm going to show you in a minute. The electrons there are becoming excited and then coming back to their normal state and then giving off light.
Yeah, I'm getting tired.
Thank you very much. You can stop now. You can stop now. Thank you. Thank you. So I just thought I'd give you an actual demonstration as to just an application of fluorescence. So I have two solutions here in the glasses. You can see they both look very similar. Visually, there isn't really a difference between the two. But when I shine this fluorescent light on them, you can see that one of them, you can see, is glowing blue. And that's because that one is capable of fluorescing, whereas this other one isn't. And do you know what this might be? Does anyone have any ideas of any solutions that we have, something we drink that might be fluorescent? So this one here is water, and you can see it doesn't fluoresce. But this one here is something that you might have maybe with some gin.
Tonic.
So you can see there are things all around us in everyday life that we may not know that actually are fluorescent. So another thing you might not be familiar with as being fluorescent is the jellyfish. This isn't real, so don't panic. And it's the jellyfish that's actually contributed greatly to scientific research. So there's one type of jellyfish that they discovered had a green fluorescent protein, which we call GFP in the lab. We managed, in the early 90s, they managed to start to use this in the lab, and it's enabled us to look at things that we wouldn't normally be able to see with a normal microscope. So an example of this is we can tag proteins that we're interested in with this protein. We can make different colours of this protein. We can tag it to the proteins we're interested in, and then we can visualise it using a fluorescent microscope. So if you look at a cell without the fluorescence, you wouldn't be able to see all these details, but now we can use the fluorescence as a tool to let us look at all the different things inside the cells. So this is actually a picture of the cytoskeleton of a cell, which is rather like your skeleton. It's kind of responsible for holding the shape, maintaining shape, and keeping it kind of rigid, a bit like our skeleton. And this is just another example of a similar thing in a different cell type, which you're very welcome to come and look at in a second. Another application that we have is that we have fluorescent dyes that we can put into cells and allow us to look at things in real time. So, for example, if we want to look at a protein and see how it moves around the cell, or if we want to look at levels of proteins increasing and decreasing, we can use special fluorescent dyes for this. So I have an example here that I can show you. So here are some heart cells that we've loaded with a fluorescent dye that is responsive to calcium levels. So as the calcium level within inside the cell increases, the fluorescence will increase, and as it decreases, the fluorescence will decrease. So it's just a way for us to monitor in real time what's actually happening within the cell.